Good day once again, welcome to Synergism International Academy, and today we're going to be treating and continuing our series on our microbial ecology, and today we're going to be looking at positive interactions. Now, if you remember, we said that we have two positive interactions. We've defined that already in our earlier lecture. There's no time to start going back into that. We said we have two types of positive interaction, and what are they? We mentioned commensalism, right? We mentioned commensalism as one of the positive interaction, and we also mentioned what? And we also mentioned our synergism, where we said that synergism here we can also refer to it as our proto-cooperation, right? Yeah, very good. So now we'll be looking at them one after the other. Now, what's commensalism? When we hear the word commensalism, what's commensalism, right? So commensalism, basically, when you want to define commensalism, you want to reflect the fact that one organism is benefiting while the other organism is neutral. It's not benefiting, neither is it suffering loss. So a commensalism association is one in which one organism benefits from the association. One organism benefits, right? One organism benefits while the other organism, now this other organism is usually the host in this case, but it's not a suffering host, I guess, because it's not suffering any loss. So while the other organism neither benefits nor loses. Very simple. Very simple. It neither benefits nor loses. So in this case here, the most important keyword you want to highlight from commensalism is the fact that I want to bring out that is one organism that is benefiting here, one organism is benefiting, and the other organism is neither benefiting nor losing. So you can say that that other organism is just neutral, within the association. Very good. Now, you see, whenever you are told to probably explain an association and interaction, especially in microbiology, do not use macro concept. Do not use macroscopic concepts as examples. For instance, now there is the common examples where people want to make use of to explain commensalism. So people want to just go into maybe remora fish and shark or whale and barnacles. Those things are macroscopic concepts. You can't bring them in as examples when you are discussing commensalism in microbial ecology. Very important. If you go do that kind of stuff, you're going to get that question wrong. So if you are answering microbiology question, to make sure you use microbial examples. So let's quickly look at some of the examples we have in commensalism that are microscopic. So we'll look at the first one. Let's probably discuss on the let's just say microflora, the microflora of skin versus the human skin. Yes. Versus the human skin. Now, one thing I want you to understand here is this. When we hear the word microflora, these are natural microorganisms you find within a biological surface. So in this case here now, the microflora dealing with are those natural organisms you find on the skin. Yes, they are always there. They are always there, naturally existing there. And the reason why they can naturally exist there is because they cause no harm. So you understand, to that organism. So here now, on the skin of humans, we find a particular microflora that we call what? That we call epidermidis. Are we there now? So you have the epidermidis, which you can, like in full, we can call it Staphylococcus epidermidis. So let me just put it here, S, where that S stands for Staphylococcus. So I'm saying Staphylococcus epidermidis. You write it in small letters, specific name, epidermidis. So this organism is found on the skin of humans. And what do they do on the skin of humans? They do three major things. They feed on sweat. And I believe they are not using your sweat for anything, right? Good. So the ability to feed on your sweat does not harm you, and neither does it benefit you. But it's benefiting them because that's their own source of food. So they feed on your sweat, and I believe you know that on your skin, or within the skin, you have your subcutaneous layer, and you have regions where you have fat, right? And those fats usually can generate oil. So good, they also feed on oil. You can see that now. So they can feed on your sweat. They can also feed on oil on top of the skin surface. which are things that do not pull harm to the human himself. So whether they feed on your sweat, are you getting now, or they don't, it is not beneficial, neither is it a loss to you, but it's beneficial to them. Do you get that now? So very, very good. So also I said that they feed on oil, right? Another thing again is that place serves as the skin will now serve as habitation for them. So they begin to inhabit those regions. So that's that for this association. Is a commensal association. You can see that there. So the microflora of the skin, which is Staphylococcus epidermidis, is benefiting from the human skin by feeding on sweat, on oil, and also utilizing the skin as habitation for their survival. That's one. Another thing again, another example again we can go into, because in your Jupeb exam you need to understand that you should be able to write. You're not just going there to go and answer questions like a secondary school student. You should be able to write. When you are listing examples of commensal association or interaction in microbiology, you don't just write microbial skin versus on the skin. No, you have to state what is that benefit that this guy is getting, and how does that show that it's not pulling any benefit or loss to that host. Alright, then another one that we will now probably talk about again is we'll look at an association of nitrosomonas and nitrobacter. Yes, and nitrobacter in the soil. Now, in the soil, we know that there are nutrients in the soil. We have ammonia in the soil, and good. We know that nitrosomonas is a bacteria that does what? Nitrosomonas is a bacteria that is doing what? That will convert. Nitrosomonas will convert what to what? It will convert ammonia by oxidation, to oxidize ammonia to form what? It will oxidize ammonia to form what we call nitrites. So once nitrosomonas have acted on ammonia, it will produce nitrites. That nitrites now, it serves as food to nitrobacter. That's the food of nitrobacter. So when it has converted ammonia to nitrite, nitrobacter on its own, nitrobacter will now pick up nitrites and feed on nitrites. So we just put here, nitrobacter feeds on nitrites. And when it feeds on nitrite, we know that the byproduct produces what? Is nitrate. But we're less concerned about that now. We are concerned about the nitrite. So it feeds on the produce of nitrosomonas. So it's just as good as nitrobacter benefits from the product of nitrosomonas, and nitrosomonas is not harmed by nitrobacter feeding on its nitrates. It's just like a waste product to it. I get what I'm saying now. So this is a commensal association. Why? Because this nitrobacter is feeding on a byproduct from nitrosomonas or a waste product of nitrosomonas, but the nitrosomonas is not benefiting from him. I get it now. Neither is he suffering from the activity of the nitrobacter. Nitrobacter feeding on the nitrates is not affecting him either. So this is a neutral guy here, and this guy here is benefiting. So it is still considered a what? It's still considered a commensal association. Now let's look at another example again. We have some association where you have two anaerobes. So you are having a facultative anaerobe and obligatory anaerobe living together or side by side. So in this case here, this thing usually pulls up a commensal kind of association. How? Now, this is it. What's an anaerobe? First of all, anaerobe are organisms that can grow in the absence of what? Oxygen. In fact, they do not want oxygen to be in the environment for them to grow. But it's not that we have two kinds of anaerobes. We have those that are facultative. A facultative anaerobe now is one that can still survive both in the presence of oxygen, I get it now, and they can also survive in the absence of oxygen. So that's why it's facultative. It is not compulsory. It has to be in an oxygen deficient environment. And that's why it's facultative anaerobe. It can still survive in the presence of what? Oxygen. But an obligatory anaerobe is a strict anaerobe, meaning that it does not want oxygen. Oxygen is harmful to it. So for this obligatory anaerobe, oxygen is harmful to them. You have to know that for obligatory anaerobe, oxygen is harmful to them. Oxygen is harmful to them. I believe that if you are thinking logically, you can quickly understand what is going on whenever the two of them are in place. For instance, now one good example of your facultative anaerobe, let's just say we want to utilize E. coli, Escherichia coli. And your obligatory anaerobe, let me put this one here so I can know. E. coli is standing as a facultative anaerobe. So the Clostridium now, let's use Clostridium. The Clostridium, we have Clostridium tetani, which causes tetanus. And then now, that one there is serving as an obligatory anaerobe. It's an obligatory anaerobe. So what happens is this: when you have this facultative anaerobe and an obligatory anaerobe in the same environment, something happens. And what's that thing that happens? The thing there is that the facultative anaerobe will be utilizing the little amount of oxygen that's available in that environment. So this guy is going to be, this guy here is facultative anaerobe. He can survive in oxygen, right? But while he is there, it will be utilizing oxygen. And when it's utilizing oxygen in that environment, what will be happening to oxygen concentration? Thus, oxygen concentration will drop. And after a period of time, that environment will become what? Oxygen deficient. Oxygen deficient. Are you seeing that now? So you now have an oxygen deficient environment. On the other hand, now that place is oxygen deficient, it now gives Clostridium tetani, which is an obligatory aerobe, ability to do what? To thrive. So that's the kind of environment your Clostridium tetani wants. And it was made available as a result of the fact that E. coli, which is a facultative anaerobe, was able to utilize all the oxygen in that environment, making that place oxygen deficient. So no more oxygen. Oxygen will no longer be able to harm this Clostridium tetani, which is an obligatory anaerobe, and now you'll be able to thrive. That means you'll be able to survive. You'll be able to survive. So that is the concept. That's the concept of your, what do you call it? Your facultative anaerobe and your obligatory anaerobe being found within the same environment. So good. We'll now go into the next one, which will be your synergism. We'll look at your synergism now. Now, for your synergism, in your synergism, something happens. And what's that? Now, in synergism, we know that two organisms or more organisms, their combined effect is found to be greater than the what? Than the sum of their individual effects. I get what I'm saying now. So you have to write that down. If they ask you to define synergism, is an association where the combined effects of two or more organisms is found to be greater than the sum of their what? Of their individual effects. That means we are trying to say in synergism 1 plus 1 is not equals to 2. Rather, 1 plus 1 can be equal to something greater than 2. Maybe it can be equal to 3. I get what I'm saying now. So that's what we're trying to say. So that means 2 of us working together is better than 2 of us working separately, but we can still work separately. So that's why we're saying that synergism is proto-cooperative. It is proto-cooperative. So it's still a proto-cooperation. Is a proto-cooperation. Proto-cooperation is almost like mutualism. Both organisms are benefiting from each other. It's just that it is not compulsory we work together. Are you getting me now? Regardless of the fact that we see good or better benefits working together, if we separate, we will still be able to thrive and survive without each other. That is proto-cooperation, which is still synergistic. I get what I'm saying now. So good. Let's quickly point out some of the biological examples that we can use when you are explaining synergism. First one I want us to look at here is mixed fermentation. The concept of mixed fermentation. Now, there's something we usually call the Kefir Culture Consortium. Kefir Culture Consortium. Yes. So the Kefir Culture Consortium, you find this thing I'm about to explain, that is this mixed fermentation in the Kefir Culture Consortium and even in yogurt. Yes. Now, basically, in yogurt and in the Kefir Culture Consortium, you will find the presence of 3 major kinds of organisms or microbes. 3 major types of microbes, you find them present there. And what are these 3 major kinds of microbes? You are going to find lab, which is your what? Which is your lactic acid bacteria. This lactic acid producing bacteria, we call it lactic acid bacteria. So you will find this guy there, lactic acid bacteria. Yes. You will also find yeast in that culture, and then you will find one that we call acetic acid bacteria, which you can just call AAB. Good. So these three guys are always present within this culture. I will explain something to you because there is a synergistic association that happens between all of them here. Now, let's go into this. First thing first, there are specific examples though of this lab and the yeast. They have specific examples. For instance, now, if you want to talk about your lab, let me write it here. Examples of your lab, because I might want to be using that. I might want to be using that for illustration. Example of the lab. For your lab, you have something that we call the lactobacillus. Are you seeing that now? You have a one that you call the lactobacillus. You have another guy again that you call the lactococcus. The lactococcus lacti, precisely. The lactococcus lacti. Are you seeing that now? But in our yeast, we also give two examples there. So in the yeast, you have one that we call the Torulasporia. You have the Torulasporia. Are you there now? So people, if you want to add this in, they have different species. We have the Delbuki, we have other ones, but most especially you have the Delbuki. So you have the Torulasporia, Delbuki, and then you also have your Saccharomyces, cerevisiae. So you have the Saccharomyces cerevisiae. Now, these two guys are different major components of your Kefir culture consortium. Yes, even this lactobacillus, we can add the name there just to complete the specific name. Since I had a specific for other ones, the specific name here is Kefiri. So the lactobacillus kefiri, you have the lactococcus lacti, you have that's for the lab. You have in your yeast, you have the Torulasporia, the Delbuki, and you have the Saccharomyces cerevisiae. All these things are very, very important for you to know. You have to be able to mention them when you are trying to give illustrations in microbiology. Now, what do these guys do? What's the major thing that they are doing? Now, the point is, your lab bacteria, any of them, whenever you find them in a given kind of environment, maybe in a region, given space, are you getting me now? What they usually do is that they utilize materials in their environment, glucose and other stuff, to produce. They utilize them to produce. Even they utilize lactic acid. They utilize milk, other kind of protein and carbohydrate as well. They utilize. They will produce something. Two major things. They will produce growth factors, and vitamins. So whenever they metabolize or they act on lactic acid or they act on other carbohydrates, they usually produce two things: growth factors and vitamins. Now, these growth factors that they produce will now be utilized by the yeast. Look at agreement, look at synergism there now. It will be utilized by the yeast. So the yeast will utilize the growth factors and the vitamin, and that will aid their own growth. So that will aid in the growth. Are you getting me now? That will aid in the growth of this yeast. So it aids in the growth of the yeast. Yes. Now, while the yeast grows, the yeast will be producing now. While it is the yeast is growing from the byproducts of this lactobacillus and lactic acid bacteria. Are you getting now? This lactic acid bacteria is also growing from utilizing these growth factors it produced. But now, how is it benefiting? It's benefiting because the primary product that it had to act on to produce growth factor was actually produced by the yeast. So when the yeast is growing, the yeast will be releasing certain kind of carbohydrates, and will be releasing lactic acid. So further metabolism, this product that will be produced by the yeast will now be utilized again by the lab. That is by these lactic acid bacteria. I get what I'm saying now. So in specific way, now if I want to use specific example, you can just tell me now that lactobacillus kefiri can utilize lactic acid or carbohydrate that was produced by yeast to form growth products and growth factors and vitamins. That growth factors and vitamins that have been produced by the lactobacillus kefiri or Lactococcus lacti will now be utilized by yeast. Yeast will feed on those their byproducts, and that will enable in the growth and development of the yeast. Which yeast are we talking about here? We are talking about either your Torulasporia, the Buki, or you are talking about Saccharomyces cerevisiae. So once they utilize those products, they will now be able to grow and produce carbohydrates, and that cycle continues. So that's one of the examples of synergism. Mixed fermentation. Very, very key. Now, let's go to another example here. I can just put it here. Number 2 examples for synergism now. Now, how about if we look at the association between the 2 organisms? Now, we're going to specifically talk about them now. Lactobacillus. Now, precisely, the species in particular that we're going to be looking at here is the Lactobacillus arabinosus. So, Lactobacillus arabinosus and your Enterococcus faecalis, they also show or demonstrate synergistic association or synergistic interaction. How? Lactobacillus arabinosus utilizes something. It utilizes phenylalanine, which is an amino acid. Phenylalanine. It requires phenylalanine to grow. So Lactobacillus arabinosus needs phenylalanine to grow. Why your Enterococcus faecalis needs folic acid to grow. Now, nature has already wedded these 2 people. Why do I say so? Because Enterococcus faecalis that needs folic acid, are you getting me now, can get its folic acid from the byproduct of Lactobacillus arabinosus. Now, Lactobacillus arabinosus produces folic acid that will be utilized or uptaken by the Enterococcus faecalis for growth. While this Lactobacillus arabinosus will utilize folic acid that will be produced by Enterococcus faecalis for growth. Both of them are benefiting from each other. That's synergistic. Now, it does not mean that they need themselves forever. Why? Because independently, they can be supplied artificial folic acids and phenylalanine respectively based on who needs who. You understand? And what did I just make you understand? Lactobacillus is the one that needs phenylalanine, and phenylalanine is produced by Enterococcus faecalis. Very, very simple. Same thing with Enterococcus faecalis needs phenylalanine, and that phenylalanine is produced by Lactobacillus arabinosus. Very, very, very simple. So we've looked at the consortium, kefiri consortium, as an example of synergism in mixed fermentation. We've looked at Lactobacillus arabinosus and Enterococcus faecalis as an example in synergism. Another one again that we might want to look at there is dental plaques. That's number 3, dental plaques. You need to know all these things. Dental plaque. You know, when you look at the teeth of some individuals, I don't want to call it specific people and set of people, but when you look at the teeth of some individuals, you find out that there are plaques forming at the roots region of their teeth, the region between the visible teeth and the gum. You see some plaques there. Now that plaque is synergistic. The bacteria that form that plaque, those things. Now, you see, anytime you see bacterial aggregates being able to attach themselves firmly to a surface, you can literally consider that stuff a biofilm, because it's bacterial attachments to a biological surface. So you can consider it a biofilm. So that strong adherence as a result of extrapyramidal kind of substances that has been released for adherence by those bacteria. Now, the point there is this: 2 colonizers are present whenever dental plaques are getting formed. 2 colonizers. Who are they? The early colonizers. You have the early colonizers, and you also have the late colonizers. Now, the early colonizers is your Streptococcus. Precisely. Now, that your Streptococcus will first of all be produced, or find its way around the mouth, and it's go and adhere itself to the teeth of that individual. When it adheres itself to the teeth of the individual family, it's a bacteria, right? So you can adhere itself making use of its glycocalyx, right? Good. Or making use of its pili. Yes, it's anchor pili. So the glycocalyx and its anchor pili usually play a vital role in that anchorage to surface to form biofilms. Good. So when it anchors itself to it, late colonizers, which is another set of organisms, will now come. What are these late colonizers? What do I call the early colonizers? You need to know those things. Early colonizers, I said we have what? Streptococcus. streptococcus. Because when you want to explain all the, maybe they tell you in an exam, explain commensalism or synergism. When you want to explain the examples, you can't just write it. You have to explain that the examples satisfies the fact that it is a synergistic association. Good. So streptococcus has adhered, are you getting now, by releasing extrapyramidal kind of substances that will help in strong adherence, and then further invasion or colonization by late colonizers will not happen, because those ones now will not be attaching themselves to streptococcus. So streptococcus is forming a surface for them to attach. I get what I'm saying there, so that they can contribute to the strength of the biofilm. I getting down why the bacteria is now, the streptococcus is now able to now find stronger anchorage to the surface, so that it can also generate or gain food nutrients from the teeth or the surface of the organism. So here now, the late colonizers usually are your actinomyces. So you have the actinomyces, and you also have the what? You also have an organism that we call the veillonella. So you have your veillonella and you have your actinomyces as late colonizers that will come and strengthen the plaque or the biofilm by attaching to the streptococcus. Are you getting now? So they are going to take part in generating nutrients from the biofilm. That's from the what do you call it? The streptococcus attachment. While the streptococcus is gaining more benefits by the strengthening of that attachment by the combination or attachment of actinomyces and veillonella on itself. It's just like maybe I'm trying to push myself on something and somebody is from my back pushing me onto that something, so I'll gain extra adherence. So that's that one for that one. Let's just add one more example, useful one. That one now is in diabetic, let's say the diabetic ulcer fits or diabetic ulcer wounds. Let's use wounds. Now, in patients that are diabetic or have ulcers, are you getting what I'm saying now? Ulcers, or maybe in their leg, you know, it's internal, not external cuts now. Now, two things are usually present within that localized region of their feet. You will find an anaerobic bacteria, and you also find an aerobic bacteria. As usual, anaerobic bacteria do not like oxygen. So what happens is that the aerobic bacteria will utilize the oxygen in that environment as usual, and when that environment oxygen has been utilized, that paves way for anaerobic. So the anaerobic bacteria now will now be able to now play its infective role within the wound. I get what I'm saying now. And when it's doing that, something usually happens. In the process of doing that, it generates fatty acid as a byproduct. Yes, when it is performing its role. That fatty acid is produced as a byproduct, usually will inhibit, it will inhibit the immunity, or you can say the defense of the host cell against anaerobic bacteria. Fatty acids usually have the ability to inhibit what they call it, defense system, and that will give the aerobic bacteria a chance to be able to also contribute or improve the infective nature of that wound. So you can see how the both of them are improving the infective nature of that wound. This one, aerobic paved way, making the environment oxygen deficient by utilizing all the oxygen. This guy now is producing fatty acids. I get him now, as it's also feeding on the host cell, is producing fatty acid. That fatty acid is inhibiting the immunity defense of the cell, of the host cell, to the aerobic bacteria. So the infectivity is what is improved. So all these things here we've talked about explains all you need to know about positive interaction in both commensalism and synergism. Take note of all the examples and do it to do extra studies. If you've not clicked or subscribed to our channel, ensure you subscribe to our channel. Also click on the notification bell so when we drop more content like this, you'll be notified. As well as feel free to ask questions on the comment sections, or if you feel that there is something you don't understand and you want us to do a video on that, do also drop that on the comment section and we'll respond to you duly.